Hi everybody. Today we are going to be starting chapter 21, which is going over amino acid metabolism. Again, I'm breaking this chapter up into two parts. Part 1 being catabolism, and then part 2 would be anabolism of amino acids. So in this lecture we are covering sections 1, 2, 3, and 4. All right, so our learning objectives today are we're going to discuss the various stages of protein degradation at the oligo level as well as the amino acid level. We'll compare and contrast differences in how dietary proteins and then proteins synthesized by our own body and cells are broken down. We'll also discuss the role of ubiquitin and how that plays into protein degradation. We'll discuss how excess nitrogen is removed from the body utilizing the urea cycle. So we're going to explain and discuss that, as well as talk about how the urea cycle is regulated. For this cycle, again, you need to know and recognize the metabolic structures as well as the enzymes involved in this cycle. We will also then sort of go through an overview of how each of the standard amino acids are degraded. However, for this, you just need to be able to categorize the amino acids by their metabolic end products, and then discuss how these metabolites are further used by the body, either through the citric acid cycle, gluconeogenesis, or fatty acid synthesis. Okay? So, with that in mind, let's get started. So, protein metabolism. So, and protein metabolism is a little different than we've seen in fatty acid and sugar metabolism, in that we involve the element nitrogen. So this is how nitrogen is transported through the body. There's no reserve storage for amino acids. So the body must continually synthesize or obtain amino acids from the diet. So making an oligomer of amino acids to form a protein, we don't do that in order to store amino acids like we do with glycogen or triglycerides. All of our proteins have some sort of other function. All right, excess dietary amino acids are not simply excreted, but metabolized into common metabolites that can be used for other processes. So in this chapter, we will examine protein degradation and deamination. We will examine how nitrogen is excreted through urea. And then we will also examine how amino acids are broken down. And in the next section, or the next lecture, we will talk about how these amino acids are synthesized. Alrighty, so protein degradation. So proteins have lifetimes that range from a few minutes to weeks or longer. Now, the degradation of proteins allows cells to do three things. 1, it allows them to store nutrients in the form of proteins and to break them down in times of metabolic need. 2, we can eliminate abnormal proteins whose accumulation would be harmful. And 3, it allows us to permit the regulation of cellular metabolism by eliminating excessive enzymes and regulatory proteins. So in table 21.1, we can see some half-lives of rat liver enzymes. And we can see we get a whole range of lifetimes. Ornithine decarboxylase is only half-life of 0.2 hours. Okay, RNA polymerase, 1.3 hours. PEP carboxylase, 5 hours. Now, some longer-lived enzymes, we have aldolase, GAPDH, cytochrome B, LDH. Remember, LDH is lactate dehydrogenase. We see that we get much longer half-life. And so what you'll notice is some of the enzymes that you may recognize as control points in other metabolic processes have very short lifetimes compared to enzymes that are not necessarily rate determining or control steps in these metabolic processes. So, protein degradation can start in lysosomes, which are these components of the cells that contain about 50 hydrolytic enzymes. The internal pH of a lysosome is 5.0. Most lysosome enzymes are inactive at cytosol pH, which is around 7.2. And there's a reason for this, and I want you to kind of ponder. You can look in this section of your chapter and read up on it, but there's a defense reason for the cell that we store lysosome pH that are only active at pH 5.0 separate from cytosol pH. And the reason is because if the lysosome were to break open and spill out these hydrolytic enzymes are non-specific, and if they were active at cytosol pH, they'd be able to eat up all the proteins in the cytosol. So in case the lysosome does bust open and these enzymes spill out into the cytosol, at the cytosol's pH, they would be inactive and would not inadvertently kill the cell. Now, in well-nourished cells, the lysosome enzymes are non-selective. However, in starvation states, biochemists have found that they do become selective to proteins released from tissues that respond to starvation through atrophy, such as the liver and kidney, and not proteins from tissues that do not react to atrophy, such as brain. What's also interesting is rheumatoid arthritis involves the extracellular release of lysosomal enzymes that can degrade proteins and cause inflammation. Extracellularly. Okay, so outside of the cell. Now, another form of protein degradation that's outside of the lysosome utilizes the protein ubiquitin. So, again, this is independent of lysosomal degradation. Ubiquitin becomes covalently attached to the protein that needs to be degraded in 3 steps. So as we can see here, first off, you notice that it involves ATP, and not only are we removing one phosphate from ATP, we're removing both the phosphates to make PPI, which remember is going to spontaneously hydrolyze to form 2 inorganic phosphates, thus releasing more energy. Alright, so we first transfer ubiquitin to E1, okay, and that's going to first activate ubiquitin. Then E2 also catalyzes another transfer of ubiquitin from E1 onto E2. Alright, so we're sort of stepwise activating ubiquitin to make it more reactive. Then E3 takes ubiquitin and transfers it to a lysine on the condemned or the protein that needs to be degraded. Okay. However, just adding one ubiquitin is not enough. Efficient degradation requires a chain of at least 4 ubiquitins tandemly linked in order to trigger degradation. All right, and so this chain, ubiquitins will be added onto the end of the preceding ubiquitin on the condemned protein. And you can have more than 4. Typically, you would have 10 to 20. So, after being tagged by a chain of ubiquitin, 26S proteasome unfolds and hydrolyzes the protein utilizing ATP. So the actual proteolytic subunits of the proteasome are the beta subunits. We also have some alpha subunits as well, and they're believed to be functioning more in the feeding of the proteins through the proteasome in order for the beta subunits to cleave at very specific sites on the protein. So beta 1 cleaves after acidic residues, beta 2 cleaves at basic residues, while beta 3 cleaves at hydrophobic residues. So after the protein has been pulled through the proteasome and cleaved at every acidic, basic, and hydrophobic residue, this leaves on average an 8 amino acid length fragments after digestion of a protein. Peptidases are then able to come in and cleave up these small fragments into individual amino acids. Ubiquitin itself is not degraded. It is released from the protein that we degraded and then returned to the cell in order to tag other proteins that need degrading. All right. So dietary protein, so protein that we consume, is degraded a little differently than proteins from the lysosome and through ubiquitin tagging. So when we eat protein, we degrade it using using a combination of gastric proteases such as pepsin, as well as pancreatic enzymes such as trypsin, chymotrypsin, and elastase. So these were discussed back in the chapter when we were talking about enzymes. So it may be worth going back and looking at those again. All right, where pepsin is sort of substrate unspecific, trypsin, chymotrypsin, and elastase are sort of more substrate specific. Okay, if you remember, chymotrypsin is going to cleave around aromatic residues. All right. Now, we degrade polypeptides first into oligopeptides and then amino acids, and then these oligopeptides and amino acids are absorbed by intestinal mucosa and are transported via the bloodstream to tissues. Further amino acid degradation occurs intracellularly. So we can see once we have amino acids in the cells, they can be degraded to carboskeleton, either sent through the citric acid cycle, sent through gluconeogenesis, becomes acetyl-CoA, which then can become fatty acids or ketone bodies, right? Okay, which can then be used for either the citric acid cycle, fatty acids, or other amino acids. Okay. The ammonia released from amino acid degradation is then sent through the citric acid cycle or used to create other amino acids that are needed for protein synthesis. All right, so when we're talking about removing nitrogen from amino acids, we call that deamination. So most amino acids are deaminated by transamination. So what we're doing is we're transferring the amino group from our amino acid to another alpha keto acid in order to make a new amino acid. Okay, so in this instance, we have just some generic amino acid, a transamination enzyme or transaminase, transfer the ammonia or the nitrogen over to alpha ketoglutarate, which makes glutamate, and then a new alpha keto acid from the original amino acid. So glutamate's amino group can then be transferred to oxaloacetate, making aspartate, as we see here. So we transfer the amino group from glutamate over to aspartate, which regenerates our alpha ketoglutarate to accept another amino group using oxaloacetate. The enzymes that govern these reactions are called aminotransferases or transaminases, and they require PLP, the coenzyme PLP, which is a derivative of vitamin B6. So the deamination of amino acids requires a Schiff-base reaction. As we can see, it's going to go in two stages. The first stage is conversion of the original amino acid to a keto acid, followed by a conversion of an alpha keto acid to an amino acid. All right, and so we kind of see that happening. The first stage, we're going in this direction, so we're transferring the amino group using PLP as our Schiff base. So PLP accepts our amino group here, in the end. And then in the reverse, in order to transfer that amino group from what is now PMP, okay, to the alpha keto acid. All right, so stage 1 is in this direction, the forward direction, while stage 2 is the reverse. So glutamate can be oxidatively deaminated. So this is the only amino acid that can actually be oxidatively deaminated, which is why it makes it unique for being able to accept ammonia or nitrogen groups from other amino acids that need to be degraded. Glutamate dehydrogenase is the enzyme that governs this, and it can remove the ammonia group from glutamate, yielding ammonia and alpha-ketoglutarate again, so that it can be used for additional transamination reactions. This enzyme is found in the mitochondria. It's one of the few enzymes that can accept either NAD+ or NADP+ as its redox coenzyme. Interestingly, it is inhibited by GTP and NADH and is activated by ADP and NAD+. So when there is a metabolic need for ATP, we activate GDH in the mitochondria to start breaking down proteins. However, when there's high levels of GTP and NADH, we inhibit this enzyme. So we are effectively inhibiting amino acid degradation. So when this happens, we have glutamate accept or donates a pair of electrons to either NAD or NADP. And this makes our transition state alpha aminoglutarate. And we say we end up with a transition state here. Using water, we hydrolyze the ammonia group off of the alpha aminoglutarate to regenerate alpha ketoglutarate. All right, and then free ammonia is released. Interestingly, I want you to notice that GTP is our inhibitor, not ATP. And so I want you to ask yourselves, why would we utilize GTP as our inhibitor versus ATP? And the reason for that is you need to go back to the citric acid cycle and look at where alpha ketoglutarate shows up in the citric acid cycle and ask yourself, why GTP and not ATP? Now, the urea cycle is how we remove the excess ammonia that is released from GDH off of glutamate. Okay? So urea is formed in the liver by enzymes of the urea cycle and then is secreted into the bloodstream. It is then sequestered in the kidneys and then excreted in urine. All right? So this is sort of our overall reaction of the urea cycle. We have our free ammonia, bicarbonate, and aspartate. We utilize 3 ATP molecules. We are going to dephosphorylize 2 of those ATPs into ADP and then 2 inorganic phosphates, but one of those ATPs are going to get dephosphorylated into AMP and PPI. And remember, PPI will spontaneously hydrolyze into 2 inorganic phosphates. All right, so effectively we are hydrolyzing 4 high-energy phosphate bonds. So this is very energy required, very high energy required reactions. In the end, we end up with a molecule of urea and then a molecule of fumarate. All right, so in our first step, Carbonyl Phosphate Synthetase is actually not technically a member of the urea cycle, but it helps prepare our ammonia for entry into the urea cycle. So what this enzyme does is it catalyzes the condensation of ammonia and bicarb to form carbamoly phosphate. And this requires 2 molecules of ATP in order to do this. So we can see first our oxygen is going to attack the phosphate oxygen bond in ATP. We release ADP. Then we're going to have ammonia attack our carbon, releasing inorganic phosphate, and now we got carbamate. Another molecule of ATP is used to transfer phosphate over to make carbonyl phosphate. Carbamoyl? Yeah. Alright. In step 2, ornithine transcarbamolase. Ornithine transcarbamolase transfers carbonyl group of carbonyl phosphate to ornithine to yield citrulline. This reaction occurs in the mitochondria, but then must be transported out to finish the urea cycle. Alright, so we can see here's where we made carbonyl phosphate. Ornithine transcarbamolase transfers this functional group over onto ornithine to make citrulline. Okay, so we're going to lose that inorganic phosphate. Argininosuccinate Synthetase then catalyzes the next step in this reaction, step 3, where it inserts a second nitrogen atom by condensation of citrulline's ureto group with an aspartate amino group. This is where we utilize that ATP molecule where we remove 2 phosphate groups to leave us with A and P and pyrophosphate, as you can see in this reaction here. So we've got citrulline is transferred over to A and P, and we release the pyrophosphate. Aspartate, the amino group of aspartate, then attacks this carbon, which will then break the bond between the A and P and citriol. We then release A and P, and now we have our end product of this reaction, argininosuccinate. Argininosuccinate. Alright, and number 4, argininosuccinase catalyzes the elimination of fumarate, leaving arginine. Fumarate is then converted to oxaloacetate, and then oxaloacetate can then be used for gluconeogenesis. Because remember, we are outside of the mitochondria. We are in the cytosol at this point. So this would be freely easily acceptable, easily usable, and accessible oxaloacetate for gluconeogenesis. Okay? So as you see here, argininosuccinase produces fumarate. The enzyme fumarase converts fumarase to malate, and then malate dehydrogenase converts to oxaloacetate. So these enzymes, you probably recognize these enzymes from the citric acid cycle. Although they exist in the citric acid cycle, these are also enzymes that are existing specifically in the cytosol as well to regenerate our oxaloacetate in the cytosol for gluconeogenesis. Okay, our other side product is arginine. Okay, so the amino acid arginine. Arginase then hydrolyzes arginine to yield urea and then regenerate the ornithine that we need to start a new cycle. Alright, so as you see here, here's our ornithine that's regenerated in the mitochondria. So ornithine in the cytosol has to be transported back into the mitochondria to start a new round of the urea cycle, and we release urea. So the hydrolysis requires a molecule of water to break that bond in step 5. So urea cycle is regulated initially at CPS, which is that very first enzyme in step 1. And this is activated by N-acetylglutamate. N-acetylglutamate is a byproduct of massive amino acid degradation. So we can see if we're getting a metabolite from amino acid degradation, then we know that we have some excess nitrogen that we need to excrete as urea. The other enzymes from steps 2 through 5 are controlled mainly on substrate concentrations. So they're basically in equilibrium. Here's our overview of the citric acid cycle. I think we are missing, however, step 1. So step 1, which is that CPS enzyme, produces the carbonyl phosphate. Remember, this is our big control point here. And then we start at step 2 in the actual cycle. Alright, so now that we have removed the ammonia, we can further break down amino acids. Amino acids can either be fully metabolized through entry into the citric acid cycle and then through the electron transport chain in order to generate CO2 and water. Or they can also be used in gluconeogenesis. Remember, we're going to get oxaloacetate as well from the urea cycle that can be used in gluconeogenesis. The standard, so, there's a number of amino acids, but we're going to focus on the standard 20 amino acids, are degraded into one of 7 metabolic intermediates, which can be divided into 2 groups. So we have glucogenic amino acids and ketogenic amino acids. So glucogenic amino acids are amino acids that can be used to make glucose, where ketogenic amino acids are used to make either fatty acids or other amino acids. Let me turn to that section real quick. I remember there was something I marked in the textbook that I wanted to mention. And I'm looking at the wrong book. This is something I need to pause and edit out. Yeah. So glucogenic amino acids are ones that are degraded down to alpha-ketoglutarate, pyruvate, succinyl-CoA, fumarate, or oxaloacetate. All right, and so therefore they are glucose precursors. So these can all be used in gluconeogenesis if we continue round in the citric acid cycle, then up into pyruvate. Ketogenic amino acids are broken down into acetyl-CoA or acetoacetate and are thus converted into fatty acids or ketone bodies. All right, so what we're going to do in the last part of this lecture is we're going to talk about all of these 7 metabolic intermediates and which amino acids are broken down to form that intermediate. I will show you the mechanism for each of them on the slide. However, I'm not going to expect you to commit every one of these mechanisms to memory. What I want you to notice is the common intermediate and which amino acids are broken down to form that intermediate. So pyruvate is the end degradation product of alanine, cysteine, glycine, serine, and threonine, as you can see here in this mechanism. So here is cysteine, alanine, serine, glycine, threonine. Okay, and so what you'll notice is that a lot of these start at different parts of the process. Threonine takes more steps to break down than glycine. So threonine gets converted to glycine. Then glycine gets converted to serine. Right, and then cysteine comes in at a different branch as well as alanine. Okay, but they all eventually become pyruvate. Oxaloacetate is formed from asparagine as well as aspartate. Okay, so these are relatively easy. All you have is removal of the ammonia group. The ammonia group of aspartate in ammonia transferase immediately becomes oxaloacetate. asparagine, we get 2 ammonia transfers. So we transfer first the R group ammonia to end up with aspartate, and then we do the same reaction. Transfer this ammonia group to alpha-ketoglutarate to get oxaloacetate. Alpha-ketoglutarate is formed by the breakdown of arginine, glutamate, glutamine, histidine, and proline. So here we see histidine, proline, arginine. They all converge down into glutamate. Glutamine, all we have to do is transfer that R group amino group to make glutamate. And then glutamate, we saw in the urea cycle, we can use GDH in order to remove the ammonia group, and we have alpha-ketoglutarate. Did I skip one? No. Succinyl-CoA is formed from isoleucine, methionine, and valine. Alright, so we've got methionine here, threonine, cysteine, and then succinyl-CoA. Alright? Acetyl-CoA and acetoacetate are formed by the degradation of leucine and lysine. Here we have lysine. We transfer that terminal amino group. And then this one is not showing leucine. I'll see if I can look that up. Alright, but here is acetoacetate, and then acetoacetate can be converted to acetyl-CoA. Oh, here's our side product of acetyl-CoA as well. Alrighty. Tryptophan is degraded to alanine and then to acetoacetate. So we have, we start out with tryptophan, go through 4 reactions to receive a side product of alanine and 3-hydroxyathanolate. Athanolate through 5 reactions is formed into alpha-ketoactivate, and then we have 7 reaction steps to form acetoacetate. Alright, and then alanine, remember, can be further degraded in order to form pyruvate. Phenylalanine and tyrosine are degraded to make fumarate and acetoacetate. So these are 2 metabolic processes that can be put either into fatty acid synthesis or the citric acid cycle. So we see we start out with phenylalanine, gets converted to tyrosine, and then we have 1, 2, 3, 4, 5 more steps to make the byproducts or the metabolite products of fumarate and acetoacetate. Alright, so that is basically everything we needed to cover for this first half of the chapter. When we meet in class next time, we're going to do the POGL activity, analyzing the urea cycle and protein degradation. Be on the lookout for the next part of the lecture, finishing up chapter 21, which will be sections, the remaining sections, which is just 6, and we may talk a little bit about nitrogen fixation, which is mostly plants, but it's worth talking about a little bit, okay? So, because that's how we obtain our nitrogen from the environment. All right, and then section 6 is on other products of amino acid metabolism. Oh, and then section 5. Section 5 is amino acid biosynthesis. So we're going to talk about that briefly. All right. So look forward to seeing you all in class. If you have any questions, again, please feel free to post it on the Facebook comment chain, your muddiest points, so that I can go over them in the beginning of next class period. All right. Y'all have a great day.